Hello, Gilad.
Hi. Yeah.
Hi. Good morning. Olá, just come, okay?
Yeah, sure. Is this working all right? Can you hear me well?
Yes.
Okay, good. Hi, Olavo. How are you doing?
Fine. Sorry for being late, but
Yeah, no problem. Do you wanna?
That we had to close one room to open the other one, so
Yeah. Okay.
we have some people signed up here already as participants, so we should be online anytime. Juliana,
Hi.
do you need você precisa de tempo para setar o streaming, etc e tal?
Isso. Tô indo para lá agora, tá?
Para lá onde?
Para colocar lá na página e depois no evento.
Beleza, então eu vou te dar. Yeah, we'll wait a couple of. So, hi everybody who's here watching. So I have another 15, 18 people watching. We've been in an internal meeting just a minute ago, so we got here a bit not late. It's 10 o'clock, but we need just like maybe 2 or 3 minutes to set up the streaming via Facebook and just for this to happen. But welcome everybody. We're very pleased to see you here. We have a reasonable audience already, and it's only 10 o'clock, so like for Brazil, 20 people on time at a meeting is actually quite a lot. We just in terms of technical issues, we'll have a couple of 20 minutes lectures, one by Gilad Feldman, one by me. You can participate either by sending chat messages through the Q&A, or you can raise your hand and we can put you in video and audio with us whenever we open for Q&A. Juliana Gad, who's the third person here without a camera, will be taking care of mediating the event. So we're just waiting for her to set up the streaming. So we're now live on Facebook as well. If you're watching via Facebook, you can participate on the Facebook chat as well. That side is much easier if you come into the Zoom link, which is provided in the events page, and do it via Zoom, because then you can show up in video as well, and it's easier for us to see the questions. So I guess we can start. Juliana, do you need some more?
Yes. No. We are live.
We're live. Okay. So we're live here. Welcome everybody. This is the first edition of the Brazilian Reproducibility Initiative debate series. We're pleased to host Dr. Gilad Feldman from Hong Kong University, who will be talking about reproducibility with us. So I'll just pull up my presentation here. Let's see where this is. There you go. So I'll just share my screen now. So welcome everybody. So this was actually meant to be a live event a while ago. So Gilad was actually in Brazil around a month ago, and this was actually a big excuse to have a barbecue. So we're really happy that we're like about to start experiments in the Brazilian Reproducibility Initiative. We wanted to get our teams together, so we're holding this crazy idea of having like this multi-center barbecue, like one in São Paulo, one in Rio, one in Porto Alegre, and we had to like give its lecture as an excuse exactly a month ago, March 24th. And of course, something went wrong in the process. The university closed like 3 days after we announced this. So, and it's probably not as much fun to have a barbecue in a screen. It's kind of like this dog television kind of thing. So we have no barbecue today. It's early in the morning anyway. But given the possibilities, we're very welcome to host Gilad online. He's not in Brazil anymore. I guess you're still in Israel at the moment. So yeah, he escaped just in time. But we're pleased to have him here. So it's nice to host as open science people, because they're very easy to present, because they have like web pages where the presentation is ready. I just kind of like copy and paste it from his own website. But Gilad is a psychologist. He's been in HKU since 2017, and he mostly focuses on judgment and decision making. But he's actually very much into like doing large scale replications and using whatever whatever kind of labor he can find, students, collaborators, colleagues, etc., to try to make this happen, which is kind of like what we do as well. It's like we're basically a group of people trying to engage people into mass replication. And I guess this is pretty much one of Gilad's main interests. He's a very dedicated open science supporter, so we're very pleased to have him here. So we have him talk for 20 minutes, maybe open up for questions. I'll talk for around 20 minutes, open up for questions, and then we just debate among everybody. We should end around maybe 11:30 or something like that. So Gilad, thanks for coming here, whatever here is, and you're welcome to speak.
Thank you. It's great to be here. I'm happy that we had the opportunity to do this, although it didn't work out in Brazil. Let me just share my screen with you. So this is my presentation. So as you can see, all the slides are downloadable, so you can just take whatever it is that's here and look at this offline. And follow while I'm talking. There's lots of hidden slides. So the original presentation takes about an hour. And this is what I was doing in different parts of Sao Paulo, hoping to do this also in Rio. That didn't work out. And I do this because I want to share with people what it is that we do in this open science, mass replication effort at University of Hong Kong with many different collaborators around the world. Share this a little bit with you. It's going to be a bit of a rush in 20 minutes. Some things I assume that you already know, open science, all that, but I will be going over this briefly. If you want the full presentation, the Sao Paulo University folks videoed, recorded everything. So you can go on this link on YouTube and watch the watch the entire 1.5 hour presentation that goes into things in more depth, covering some personal stories and things about what happened in my career that led me to do this sort of thing. Generally, the message that I have when I talk to people about open science is that we are in a process of figuring out what is going on with our science. And there are some signs that things are not as good as we have hoped with science. Some have called this a crisis, a replication, reproducibility crisis. And my personal takeaway from this, which is what I'm trying to talk to people about, is that we need to do a credibility revolution. And we need to do this fast. We can't take our time with this. And I think the coronavirus and the current crisis around the world has led us to this conclusion a lot faster than before. So we need open science in order to get to the right conclusions and solutions to address this crisis. So we must improve the way that we do research. There are all sorts of things that we've been doing wrong in our universities and academia. We also have to change the way that we teach, and I'm going to talk about how we did this at University of Hong Kong. We really have to start working together. If any small lab will try to address the coronavirus by itself, we will not solve this. But if we mass mobilize everybody together, collaborating efforts in different labs around the world, there is a chance that we'll be able to solve this in time without mass casualties beyond what is happening right now. And my take on this, and I think this is something that has not happened yet, is to mass mobilize students, how to work with students, undergraduates, as early as second year to address this crisis. There's a timeline that I usually show about what happened, when did this start. So I date this back in my career to 2011, although there are signs in 2005 with seminal paper about how most published findings are false. But for me, it started in 2011. There's all sorts of different efforts that came out and different things in this timeline that were big events. But it took us about 2011 to 2015 to kind of mass mobilize researchers to try and start running replications. So in order to validate, to assess what is going on with our science, we wanted to try and do as many replications as we could. So you can see over here, there's many labs number 1, many labs number 3. It took 3 more years in 2013 to do many labs 2 to get it published. But finally, 2015, 2016, there was the first signs, like massive, big signs that some things are wrong. A lot of big findings that we communicated to the public, like the power posing over here, and ego depletion and social priming, failed to replicate in psychology. And I took this as a sign that something is very wrong. And some of these stories, like the self-control as a limited resource, is something that I find specifically for me important, because I was involved in a lab that led this effort, this theory. But I think that the main change came in 2015 with the science paper, where they found, for us, very disappointing findings that only 36% of all attempted replications actually succeeded. And in those, the effect size was about half of what the original was. So if the published findings were about an average of a correlation of 0.4, then in the replication it was 0.2. And then since 2015, we've had a lot of RRR, registered replication reports, where a lot of different labs from around the world come together in order to try and assess what is going on with our science. This is the one that I mentioned before about ego depletion, self-control as a limited resource, that I feel connected to, specifically found no effect whatsoever. And this is the tweet that summarizes everything, with an effect size of Cohen's d of 0.08. Which is tiny. So if you do a power analysis, you would need something around 5,000 participants to test this. But since then, we've had all sorts of mass replication efforts. So this is 13 out of 21 psychology papers in Nature, in Science. Only 13 out of 21 replicated, and the effect size was much weaker than the ones published. And this one here, the many labs too, that finally got published in 2018, we're talking about 14 out of the 28. A very impressive project with a lot of labs collaborating, many different samples, 7,000 participants, disappointing findings. If we summarize all this together, to put this in perspective, it comes together with these mass replication efforts. The replication rate in social psychology is about 47%. We've also been doing a lot of registered replication reports for many other things, and a lot of failures to replicate, especially things in terms of social priming, things about embodiment. But the good news is that some things are replicable. These are some signs that this is happening. For some things, JDM, judgment decision making, which is something that I'm personally focused on in the last 2-3 years, seems to be doing fairly better than most. So many people at the beginning said, perhaps this is a problem with psychology. If we didn't expect much in psychology, so of course, if we'll test psychology replicability, reproducibility problem, then we'll find these disappointing findings. But since 2015, we've started doing this in other places. Medicine has been doing this for a while and then stopped, and now started again. And then these big headlines started to come out, saying that this is not just limited to psychology. This is happening in many other disciplines, like chemical research, economics, cancer research, and so forth. I try to summarize all these findings. You can go on these slides and have a look. I just want to say that this is not a problem in psychology. Also, the hard sciences, the exact sciences, have some issues. These are preliminary findings. These are initial replication evidence, because we haven't been doing replications enough, definitely not in the hard sciences, where things are proprietary, there's patents, there's all sorts of incentives not to share information. So if we look at the hard sciences, actually psychology seems to be doing better than many of those. So the rates are very, very disappointing, very low. And I think the biggest one here is cancer biology, where in 32 out of the 50, they weren't even able to reproduce the material, the process, what is needed to be done about this. So only 18 out of 50 could start, and out of those, 14 have finished so far. Only 9 out of those was replicated. So seems like there's some issue here. So at some point, Nature decided to do a survey and ask people, do you think there's a crisis? And about 90% said that there's either a significant crisis, 50%, or 38%, a slight crisis. So there seems to be some consensus that something is wrong, and we need to look at this more carefully. And the way to look at this is to do a lot of replications and see what works and what doesn't. And while we're doing replications, figure out what it is that isn't working very well in science. So for a while now, I've been going around and talking about this this process, this open science issue, the credibility revolution, but there hasn't been any good resource for that. So when my students ask me, so where can we learn about this? We want to read about this. I gave them some blog posts, I gave them some podcasts, I gave them some videos online, but there wasn't really a book to summarize this whole thing. So in the last semester, I used open science collaborative Google Docs, where I invited my students to help me write this. So we separated the chapters, and a group of 6 students got each chapter. And then the TAs gave some feedback, and people came from online. And finally, there's a book of about 200 pages about what is the open science credibility revolution process, what is the replication crisis. So if you want to know more about this, you're welcome to go in here and have a look and read a little bit more about that. And you're invited to also collaborate on this. So everything that we do, everything that I'm showing you here is something that you can go in and contribute. So if you go in and edit, add your name, and when we submit this somewhere, you will be a co-author. So everything is collaborative. My summary about this very short interview of the crisis is that I'm convinced that there's a crisis. So there's something that we need to look at very carefully over here. But I get a lot of pushback on this, especially as I communicate with people whose work we try and replicate. A lot of senior top scholars in the field have some issues with the open science movement or the need for replications or why this should be published. So the way that I summarize this whole thing, that I'm convinced that there's a crisis. It's okay that if you're not, but at the very least know what is happening. Follow the articles that are coming out in the mass replication efforts and evaluate the findings to see if this is in line with your expectations. When I started my journey, I'm only a third-year assistant professor at the University of Hong Kong. I was a postdoc, and I started this open science journey when I was a postdoc in the Netherlands. They seem to be very dedicated to the open science movement, following some disappointments, especially a big fraud case, Diederik Stapel. So they took it upon themselves. They took responsibility in leading an open science movement. So I learned a lot of things while in the Netherlands, and I set some principles for myself, which is what I aim to do in my own research. So this is where I think we're headed. We need to emphasize trustworthiness, reproducibility, and replicability. And the way that we do this is following all these principles. So we need to look at effect sizes rather than this p-value, null hypothesis significance testing criteria of 0.05. We do a lot more replications, doing registered reports and pre-registrations. I think that the most important one here is that we need full transparency, not only sharing all the data sets, but we also need the code in order to figure out what's going on, and it needs to be very well documented, and it needs to be one that anybody can run and follow what it is that we did. But also, every decision that we've made from the first time that we ever thought about an issue up until when we publish it, everything needs to be documented about all the decisions, because there's so much flexibility when we do research about how to analyze, how to think, how to design the experiments or the studies. So I document everything, I share everything. If you go on my website, you'll see each project, what status it's in, and you can go and check the open science framework in order to see what's going on with this. This is a slide that I really like from Simin Vazir about the difference between incredible, we have these amazing findings that we publish in Nature and Science, moving from this incredible where papers are advertisements and only few people get all the glory and people do things in their own lab, and there's blind faith in this peer review, this magical peer review process, moving to credibility here on the left. So being more transparent, doing pre-registrations, being very open about our conflicts of interest, and giving credit to contributions, allowing people to replicate our work, just trying to minimize errors in published research. So this is kind of like what led me to my beginning of my position at University of Hong Kong. The main insights for me was that we need to change the way that we do research. But if we do this on our own, if it's just us professors, it's going to take us a very long time. So we need to start working with our students. So we have these big classes. I teach classes of up to 100, sometimes 150. We have even bigger classes than that. So as early as second year, we can work with our students. We can train them, even if they come into our classroom not knowing a lot of statistics and not knowing anything about open science. I have taken the opportunity given to me by University of Hong Kong to try and in one semester get them from almost knowing nothing about science to completing a whole project of doing a replication of a classic finding in judgment and decision making. So I took some ideas from other projects that are happening. There's this CREP and the Psychological Science Accelerator. You can follow these links and read a little bit more about that. Also, Sanjay here has this really interesting syllabus about how everything needs to be fixed, psychology, significance testing. So I go with my students every week. We talk about different things that need to be changed. What is the current situation in science and what needs to be improved? But the main principles are that students lead the way. Right now, most of our books contain a lot of findings that we cannot replicate, and we can work with them in order to try and figure out what is actually replicable, trustworthy, and what isn't. I'm going to very briefly go over the process of how we do replications. So each replication target, each article that we want to replicate is given to two teams of two students working independently. And these teams are working on the same target, but then at some point, so they design a Qualtrics survey. So this is kind of like an online experiment that can be run. We randomly generate a data set from Qualtrics, and then they, in advance, do a data analysis plan. They write a pre-registration report, and then they peer review one another. So the two teams switch, and then they look at what the other team has done. They also get external feedback, both from the TAs, myself, and other people, other scholars. So I open everything up on Twitter. Then we do, then they do a revision. They give this to me. I do all the pre-registrations of the Open Science Framework. I do all the data collections online using Amazon Mechanical Turk and Prolific. And then once again, they do data analysis. They peer review one another. They write the final reports. They get external peer reviews, open peer review on Twitter. And then finally, they submit a revised final report, which is supposed to be a manuscript that can be taken almost as is and submitted to a journal. We don't submit this just yet, but some of them are very, very high quality. There's one more step missing, and this is the step where I invite you to join us on this journey. But I just want to say that these reports here at the end of the semester are already quality that includes everything from, is introduction, methods, results, discussion, full transparency, everything included. And we share this with the world. We put this on the Open Science Framework. If you want to know how I do this, it's very simple, but we don't have enough time to go into this. Everything that I do in the spirit of open science is available to you on the Open Science Framework. So you can go here, click on these links, and see what it is that we do in our process. In our replication effort is slightly higher than in social psychology, so judgment decision making. Our replicability rate is about 70%, and we ran 57 of those. So if you think about this, in 3 semesters, using our work with my students at University of Hong Kong, we were able to achieve 57 replications, making this one of the largest social psychology replication efforts. So there's a lot of things that you can do with this. And if you expected very little of students and Amazon Mechanical Turk online data collection, then I think 70% speaks not only to the replicability of judgment decision making, but also what it is that you can do with students. We're doing a lot of other things This is an open invitation to you. So we develop resources to help other people, not just ourselves, conduct replication. So if you want to conduct a replication, we have a very detailed guide over here. We have a collaborative guide on how to do extensions. We have a collaborative guide on how to do R, Jamovi, JASP, how to calculate effect sizes, how to work with the Qualtrics. And all of these are open to you to come and co-author with us. So if you contribute anything, just put in your name, and when we submit this to the journal, you'll be a co-author. We also have some really great templates that you can take in and use, and this really helps students go with structure, with instructions, step by step on what it is that a report needs to be done. I won't go into this, but if you want to see how good these reports are, you're welcome to evaluate these. Everything is open, everything is shared. Just follow the links. They did a really good job with this. And this is the open invitation to you, almost the last slide. If you want to join us, what it is that we do is that we invite other scholars, typically early career researchers, end of PhD, postdoc, or beginning of assistant professor, to come and take our students' reports, verify these, add some analysis, make sure that the introduction and the discussion are solid, and then submit this to the journal with you as the lead author. We right now have about 15 collaborators. As you know, we have 57 projects completed, so there's 30 more that are waiting for you to come in and take the lead. And some of those are already published. So we are able to publish these replications in really good journals. So this is from SPPS, and JSP. These are good journals in our field. In metapsychology, this is a new journal, completely open, peer reviewed. As you can see, these underlined people are my students, and these are the early career researchers that took the lead. We also have a lot of preprints, so you can follow all that. We've been getting some media attention, giving us some compliments about how this is a good evidence that rigorous replications can be run by researchers who do not yet have a PhD. So we don't need scholars or only scholars to do these replications. We can work with our students on that. Not only replications, we do other things like assessments. So if you want to look at what other people have done in their research, so we have a template for that, and that's a whole different other project that we can talk about later. But we can mass mobilize students not only to do replications, but also to evaluate what it is that's out there, evaluate people's pre-registration, their code, their data, and what it is that they shared. So you can join us in all sorts of different ways, collaborate with us on manuals, integrate this into your courses. Bottom line is, tell me how I can help you to do open science and promote replications, reproducibility in your field. Everything that I just talked about is summarized in this page, so you can go on this, click on there, on the link over here, and it will give you a long list of all the resources, everything that's open, a lot of explanations, and all my contact details are up over here. So if you want to have a more personal discussion about this, you're welcome to contact me at any time. Thank you. I think you need to unmute the microphone.
Yes, sorry. Yes, thank you. This was really great. If anybody has a very pressing question concerning specific topics within Gilad's lecture, you can do it now. Otherwise, if there's very general questions which might be interesting for me to pitch in as well, you can also wait for my 20-minute lecture here, and we can do a big Q&A afterwards. But if anybody has a more specific question, I think we can open right now. You can either raise your hand or do it via chat. If not, I think I'll just move on. You can also do, you can ask questions while we speak via the chat button below. So we just might not answer them immediately, but they will be stuck there once we're done here. Clarissa Nascimento. As from your experience, how open other areas in biology are for open science projects? That might be better for us to talk later, because I think that's one big question. Like, psychology is doing much better than we are, but you can say.
Yeah, so I don't know a lot about this aside from the little that I see in like meta science conferences and what I follow online on Twitter. So there seems to be some projects right now, including this cancer reproducibility project, and it seems like things are not going very smoothly. There's lots of issues, especially with reproducibility, with just understanding what it is that these scholars did in their lab, because I think by nature, by default, we're not open about the way that we do biology research. So there's a lot of, this actually surprised me, because I also thought that the psychology was behind on this, but I understand now all the challenges about being open about all this, especially in a very competitive market where you need to capitalize your ideas at the end. But yeah, this is, I'm excited about working with people in biology, because we don't do this a lot. Just try and see how we can like leverage what it is that we're doing in psychology, leverage what's happening in biology in order to create something that's shared, moving from just psychology to more meta science.
Yeah, of course we can't do it over Mechanical Turk, right? So I mean, we have some big disadvantages. It's more expensive. So I'll go into that. Maybe I'll just use this as the hook to my lecture. So let me share my screen here. Let me just find, yeah, so here I am. So moving on, we're trying to then switch gears from psychology to biology, but acknowledging that psychology is ahead on this. Of course, they have some advantages. A lot of it is like cheaper to do. You can do it online, but they have also moved ahead of us. I mean, they've been discussing this very, very intensively over a decade at least now. And I think we're getting there, but we're getting there late. So I'll skip the part of why reproducibility is important and that we have a problem. I think Gilad has talked about that. I'll just you can take his word for this. But I'll just point out the fact that we have very, very little data on this in biology. We have these papers from pharma from around almost 10 years ago now, which say, well, we can reproduce 11% or 21% of what's published in academic papers. But those are like very out of the blue numbers. These are industry numbers. We don't have, this is very bad in terms of open science. We don't know the papers, etc. That said, they made an impact. I think they got people talking. The closest we have to a more like fully open science project is a reproducibility project, Cancer Biology, by Tim Arrington at the Center of Open Science, which has run into a lot of trouble. I mean, they have replicated 15 studies, and this is probably like the best data set we have. And that's like 15 studies, highly selected, very highly cited within a specific field. So I mean, we don't really have a lot of data on how much of the biology literature actually reproduces, and it's hard to get. Like the RPCB cost around $1 million. Like each paper replicated here is probably as expensive as the whole of the 57 replications that Gilad showed us. Seriously, so it's a challenge, right? And maybe it's money we should be setting up to spend, but it's not particularly in the culture of funding agencies or institutions. I mean, if you do, and we don't have numbers in an international setting, and much less at a local setting. And again, I think it's important to evaluate reproducibility at a local setting. We know that it's probably very connected to like how science is incentivized, how the scientific environment functions, and of course that is very local. I mean, the kind of thing you incentivize people to do have to do like national funding agencies, local state funding agencies. And what we have seen in Brazil over the last 20 years, maybe more, it's like explosive growth. Of course, now we're tapering. This goes up to 2015. Now we're going through a bad slump. This is probably falling a bit. But again, like from the 90s to 2015, at least, we had a very large growth, and people have been optimistic about it. I mean, a lot of, in terms of like published papers, grad students, everything grew very fast. And even when people manifest worry about it, it's mostly about, okay, oh, so yeah, we're growing, we're publishing more, but we're not getting cited much more. The impact is about the same, so we're doing more of the same. It's aligned with science, whatever, which is important. But I mean, there's a much more pressing question here, is that whether this is actually true. I mean, if you're just like growing our scientific output in terms of like junk data and things that do not reproduce, this is actually not good news at all. I mean, it could be a cancer in terms of like unregulated growth, which ends up into bad science. That said, again, that is not exactly, it's not a very popular thing to evaluate your own science. I mean, funding agencies are not really typically very willing to do it, institutions are not. But I guess, again, if this is out of the scope of funding agencies, not only in Brazil, but elsewhere in the world, I think it should become the duty of the scientific community. And I think it's telling that most big replication projects have been grassroots efforts, and most have actually been carried out with private funding. That's the case with the COS projects you had talked about. And we also managed to get funding from this from a private fund in Brazil, which is the Serra Pilheira Institute. Serra Pilheira Institute is a very young founder. We're one of their first big projects, and we're very happy to have this opportunity to provide some kind of a bird's eye view of particular fields within Brazilian biomedical science. So the Brazilian Reproducibility Initiative is a multi-center effort to estimate the reproducibility of Brazilian biomedical science in some given field. So our aim is to reproduce between 60 and 100 experiments from Brazilian articles over the last 20 years using 3 to 5 common lab methods in multiple labs. So this is more or less how our flowchart goes. I'll briefly go into this many steps here. So we started out with a very simple systematic review just to see like what methods and what models are more commonly used in Brazilian science. So as to know in what areas can we actually expect to find enough people to do mass replication. So most people use rodent cell lines. There's some common methods. So we selected 10 common lab methods for further evaluation, which we found in a lot of Brazilian papers. So Western blotting, PCR, immunohistochemistry, common stuff. And we set out an open call a couple of years ago for any labs working within these methods who wanted to reproduce experiments in Brazilian science to join. Of course, we would, through our funders, fund the experiments and people would come in with their time. And we did very intensive outreach for labs, both going to like scientific events in Brazil, going to local universities, doing a lot of social media outreach. We had this call open for around 3 months, I think August through November of 2018. And we're actually very successful in recruiting labs, like surprisingly successful. We were expecting maybe to get like 30 to 50 labs interested. We got like 71 labs to sign up, which I mean, was very surprising to us all over the country, really. I mean, most of the Brazilian states have a lab that had at least signed up for the initiative. We couldn't take them all because we had to select some methods, but we're currently around 65 labs strong, which is a lot. Most, of course, are in the Southeast, but I mean, that's the distribution of Brazilian science. Most people are young. Most got their PhDs after 2000. Well, that's not that young. I mean, that puts me into the young category. But I mean, we have a bunch of young people who are interested in reproducibility. I think this is awesome, and this is the best news of the project so far. So this is what we currently look like. We do this. We just had an online meeting with like around like 50 or 60 participants at the same time. We never got everybody together in the same place because that costs money, but we do this online meetings every 3 months or something like that. And we currently have 65 participating labs. We have the first wave of experiments starting now. So we selected 3 methods for this initial wave. We would like to add 1 or 2 more. We're not sure we have the money for that. So like this is what's guaranteed to happen at the moment. So we have 20 experiments with each method, with the MTT assay, which measures self-viability in culture, RT-PCR, which measures mRNA expression either in tissue or in culture, and the elevated plasmase, which is a rodent model of anxiety. So we're taking experiments using cell lines, rats, or mice as biological models. And we selected experiments out of a random sample of 30,000 articles in Life Sciences Journal with a list half of authors, including the corresponding one based in Brazil. We did this like via Sci-Hub. We downloaded a lot of PDFs. And then we selected through full text mining and manual screening 20 individual experiments using each method, which fit this criteria here. So they had to be a quantitative comparison between 2 groups. And importantly, we're just taking 1 experiment out of every paper. So we're not trying to replicate the whole paper as the Reproducibility Project Cancer Biology, for example. We're taking individual experiments. We do consider experiments as the building block of science. Experiments are quantitative. They have a specific effect size. It's much easier to do statistics over experiments than over papers, which have qualitative subjective conclusions. So we're taking experiments which compare to groups which have a statistically significant difference. That's important for us to calculate sample size using the method of interest, which are important enough to be mentioned in the abstract of the paper. So we don't want to get like this negative control or something like that. And done with commercially available materials because we have to buy stuff to do this again, right? And each experiment will be replicated in 3 different labs from our network in a blinded fashion with bias control measures. We're calculating sample size to provide 95% power for each individual replication to detect the original difference. We give a very large power for the 3 of them together. Of course, we know the effect sizes are inflated probably in the publication, as Gilad told us about already, so we're trying to overshoot so as to have enough power. Importantly, there's another inclusion criteria here, which is we can only replicate experiments in which we have our 20-something labs in each method have enough expertise for us to find 3 labs that can do it. So of course, like this is filtered through a feasibility thing. We usually do not take very specialized, very experiments because we won't have 3 labs being able to do them. So I mean, it's not a real random sample of Brazilian science, but it's like a random sample enriched in simple, cheap, and feasible experiments that a lot of people can do. And we're aiming for what we call naturalistic reproducibility. So we're trying to have each lab independently make their own replication decisions in terms, especially of what's not described in the original protocol. So you do get this example. So animals and housing, bulbs, mice were housed in pathogen-free conditions and fed a diet of autoclaved food and water. So this is what is in the paper, very little. So we don't really know the age of the animals, the sex of the animals, the weight of the animals, the temperature in the animal facility, whatever. But the idea is, if this is not mentioned in the paper, each lab should try to do this independently. We want to have the best fitting of what the replication looks like if each lab is trying to reproduce this just from using the data from the original paper. So they have to make these decisions. They receive like big forms with big gaps, and each lab had to fill this independently in order to get a sense. We don't want to over standardize. If we just like took a consensus decision of, oh, this is what we do, we're probably underestimating the real inter-lab variation of people trying to do this, which is important. I'll get to this in data analysis. We had a round of internal peer review, as Gilad had in his project as well. So these initial protocols went through a round of peer review by an independent lab working the same method and the coordinating team, which aimed to review interpretations from what the paper said and what people actually chose to do, detecting methodological problems, making recommendations, suggesting necessary controls, both positive and negative. So we're trying to make the protocol stronger through this internal peer review. Again, the peer review is independent, not fully because the coordinating team looks at all of them, but each independent protocol gets reviewed by an independent lab. So we're trying to keep this variation. And of course, the replicating lab also always has the final word in, okay, no, we want to do this. I mean, we can recommend something different, but they have the final word on how they run the experiment. In the meantime, we also did a prediction markets surveys kind of thing. So we're interested in whether researchers can predict what results will be reproducible, what will not. So we did this with the help of Anna Dreber in Stockholm, Domenic Veganola, and other people. Late last year, early this year, we had 2 rounds of surveys and markets in December and February. They were open to any researcher with experience in experimental research. Of course, like the vast majority here are life sciences researchers, but we had 1 or 2 people from different areas, which is interesting. We had individual surveys where we had to predict the likelihood of each experiment replicating in a qualitative sense, like whether this will find a significant effect in the same direction, and in a quantitative sense. So do we expect the effect size to be smaller, greater, like or the same as the original one? People also had to fill in their reasons for making predictions. So we're interested in like what researchers actually take into account when they do these predictions. So they did this via surveys, and after the survey, they got invited to a prediction market where it pretty much works like a stock market. You can buy stocks of an experiment replicating or not. This prices float over time, and so people can actually calibrate their predictions by looking at what other people are predicting, and this, according to economists, might have some effect in terms of like the wisdom of crowds functioning. We had over 2,000 predictions with 57 participants participating. So what we've done so far is pretty much that. We spent some time in selecting experiments. We spent some time in developing protocols and reviewing protocols. We did this prediction surveys. We have consolidated our protocols between late last year and early this year. So we're finally like, this took a lot longer than we thought. We thought we'd be starting experiments maybe like in the middle of last year, but it took at least 6 months longer than we expected. It's very hard to keep everybody on schedule. It's a lot of labs. But we're pretty much ready. So the blue ones are the ones that are ready to go. The purple ones are going through ethical review, but the protocols are ready as well. So like a few are still missing, but March was to be the month where we like start buying stuff and signing to labs and see the experiments happen. But then like the virus ate my project, and it's quite ironic. Just as we're ready to start, every lab, like 85% of our labs are closed down now. 15% are functioning with the street. So we're probably not starting out very much at the moment. We'll have to wait till this weird time passes to see the experiments happen. But they're pretty much ready to go, which is the good news. After they're ready, we proceed to data analysis. So our idea is that our main outcome for application success will be the original study's effect size being within the 95% prediction interval defined by meta-analysis of the replications. And this is interesting because using multiple labs can actually allow you to assess inter-lab variation, like real inter-lab variation from methodological differences, condition differences, which is not due to bias. And I think this is very important. We have very little data of how well data replicates across labs. So we can have like this. So these are three possible scenarios you can have. So this is the effect size here in the x-axis. So you can have three replications actually agreeing very much among themselves and the original study being within this. So I mean, this is what we call a successful replication. You can have our three replications again agreeing among themselves and the original study being way out of bounds. And this is where we say, well, this probably has some kind of problem because even in three different labs, nobody gets close to this. But you can have this. I mean, you can possibly have replications that even doing it blindly, theoretically unbiased, we still get different results because people do different methodological decisions because conditions vary from one lab to the other. And then it's actually very hard to say, to judge the original finding. I mean, if we don't agree among ourselves, this may be just due to, I mean, this varies a lot from lab to lab, depending on small methods differences. And again, this might be the case in a great many experiments, and it's interesting to know as well. We're also interested in comparing this to more traditional reproducibility measures, such as, okay, do we find a significant effect in the same direction? There's effect size within the confidence interval, etc. And we're doing some modeling work, but particularly Kleber, which is in the coordinating committee, to see how this stack up. We have done some Shiny apps to perform simulations of different scenarios to see how well these reproducibility measures measure against each other. And of course, once we have a set of replicated and non-replicated findings, we want to evaluate whether we can predict what reproduces from the original articles. So we want to see whether reproducibility depends on the experimental model method, depends on the original effect size, significance, p-values, depends on the impact measured by citation or impact factor of the original publication, whether it's getting better or getting worse over a 20-year period, whether it depends on features of the institution of the investigator, and whether it can be predicted by other researchers. Of course, we don't know the variation in this, so we don't know, we're not sure we have the power to see all of this, but we'll at least try to perform these correlations. Ultimately, I think the project is interesting because we learn a lot from it. Like even before anything happens, we don't have any results, but I think like we're learning every day from the experience because I mean, we're not used to do this large scale collaborative projects in biology, and it's fascinating what you learn from it. One thing we've learned is actually possible to do. So like engaging a large number of researchers in a collaborative project is feasible. There's a lot of people out there who are interested. Of course, they have to be funded, but people are willing to put their time into this. They think it's a valid effort, and I think the feedback we get from our communities is quite good. That said, keeping everybody together in the same schedule is not feasible at all. Like labs have their own ways to work. They have their own rhythms. There's all kinds of problems happening. So, I mean, we started out like, okay, so like everybody finishes this until then, and then we go to the next step and people have 2 months to finish this, and it just doesn't work with 65 labs. So like people have to take their time because people are different and conditions are different, and you have to be very respectful of that. It's always a hard balance between democracy and centralizing. We're very centralizing, I confess. Like we started out with a protocol that was pretty much devised by us, and like the main idea of the project was pretty much centralized. That said, in terms of like experimental decisions, we try to use our collaborators' expertise as much as possible. Final decisions on the protocols are always on the replicating lab side. We're concerned of maintaining the project's identity. So like the main goals are this, but like you're the guys that actually run the lab and know how to do this experience. We're actually working with techniques which we in the coordinating team really do not have expertise with. So it's a challenge to balance this as well. And the second thing we learned, like biologists are just not, and that includes ourselves, are just not used to the logic of the pre-registered confirmatory experiment, the kind of like register report thing that Gilad has mentioned. We have no culture of that. Most of what we do is very, very done in an exploratory fashion, which is not completely wrong. I mean, it's okay to have exploratory science. I think it's interesting. A lot of good insights come from this. But if you want to move to confirmation, as people have argued, we have to create a methodological framework for this to happen. Inclusion criteria for experiments. We have recently written about this. And things that people are just not used to. I mean, everybody, when we started out, like, oh, do you want to suggest any controls? Like, everybody has controls to suggest, but like, how are you going to use this? Like, what criteria you use? Like, when to say that this is valid or not? This can go on. People are just not used to like presetting it. So actually, that might create bias more because you can interpret the control in any way. So like, we have to learn some stuff. We have to like figure out the best way to do this, and this is not easy. I mean, again, biology, basic biology, runs in a different way from like a clinical trial that is very much planned from the start, or from a large imaging experiments, or from what can be done in psychology. Like, protocols are just on the go for many reasons which are valid. And it's not easy, but I think it's a very, very important question. How do you actually adapt this confirmatory science frameworks to experiments in basic biomedical science? This is it. We're also very present online. We're not as good as Gilad in sharing everything, but we share a lot. Maybe more chaotically, we have a website. We have an OSF page as well, where you can see protocols and details of the project. We have a feature article that describes the main outcome, the main outlook of it in eLife last year. We have been covered by a lot of media, and we are pretty active on social networks. So you can follow us on Facebook, Twitter, or you can get in touch via email. And well, this is about it. We're open for questions. I would like to thank the organizing committee, present and past members, which are truly awesome. And I would like to thank our funder. And fundamentally, I'd like to thank the over 150 researchers around the country who are currently engaged in the project. We have 60-something labs at the moment. They're very, very, very spread out across Brazil. And this is very, I mean, this is truly, truly, truly a pleasure for us to be working with all these people and to learn from all of them and to see whether this can actually happen as we thought it could. So thank you very much. And we're officially open for questions. We have some people contributing via chat. So I think we can take these. So Jorge Quilford is talking about the hydroxychloroquine story and debates on the subject, and whether Jorge, do you want to do this question? It's a long one. Do you want to do this on video? As but I think the idea here is whether the attempt for scientific outreach might be connected to the reproducibility crisis in biomedical science. I mean, I guess you're talking, we've had very specific local examples of, there was a paper describing a hydroxychloroquine plus azithromycin in very early flu symptoms that actually circulated via social networks and WhatsApp and whatever, even before it was posted as a preprint. So there's a lot of debate going on in social media, like outside of the scientific sphere, on science, on hard science these days, because of the pandemic and everything. So do you think this is somehow connected to the reproducibility crisis, or is this just a new thing that goes on top of it? I think you're muted.
Yeah, to me, this whole, so I think coronavirus crisis really showed us that we have a problem in the way that we communicate science. It's not only that there's something problematic about the way that we do science or the way that we articulate science to one another in publications, but there's also a problem in what happens between a publication or preprint to how things are communicated to the public. And as a citizen, as a layperson, how do I make sense of what is real and what is not? And especially when there's lots of different findings and everything is preliminary and you have preprints and there's a lot of noise, how do you aggregate stuff? How do you know what is, the aggregation of scientific evidence? And we don't have good platforms for this. So Open Science Framework has only existed for about 5-6 years, revolutionary in the sense that now we can share and open things up. But we also need a way to make sense of these findings. So now there are all sorts of initiatives. I know the ones in psychology, like Curate Science, where people update, not only upload their preprints, but also in a way of structuring the data. What are the effect sizes, the confidence intervals, and then all the parameters, just like you showed in your slides, what this makes this study different than others in all sorts of things, and then the aggregation of things so that people can take something out of that and come to some conclusion. But I think there's definitely a crisis in the way that we communicate. And also we need to figure out how we take what it is that we do and bring this to the public so that we don't lose their trust again in communicating things that are false.
Yeah, I fully agree. And I think it's really a crisis of we don't have anything above this was published in a peer-reviewed publication as a way of, okay, this is solid science. And being a peer-reviewed publication doesn't really mean anything in terms of like, well, or very little in terms of quality control. And of course, there's things we know, right? I mean, we know that if you apply insulin to somebody, glucose goes down. We know the Earth is round. We know the planet is warming up and it's probably anthropogenic. So there's things that we're very certain about. There's things we're like almost certain about, and there's things that are published but are still very, very preliminary. And I don't think the scientific community has found a way to differentiate those. I think like clinical medicine has attempts at it. So they have like guidelines. They have a much more established tradition of like meta-analysis of like grading evidence. So I mean, that might be the best example I know, which is like closest to biology. But again, I think we do a bad job in terms of like curating what is published. And like, okay, we have a stamp that like, this is very, 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 very likely to be true, which has to be greater than just published in a peer-reviewed publication. We don't really do that job in terms of like grading what is published and what is like very likely to be true, what is still doubtful.
Right. Well, one of the tasks that I gave my students was to try and assess the quality of both a study and a literature. And typically when we as scholars try to see, is there an effect, isn't there an effect, we used to go to our meta-analysis. But if you think about it, a meta-analysis is published once in 10, 20 years, after you have a number of studies. And then now we understand the meta-analysis typically covers what is published, and it's very difficult to get people to open up their file drawer and things that are not published. So we've developed over the last 10 years all sorts of measures in order to assess publication bias. But still, if we need to wait 5, 10, 20 years for meta-analysis to be updated, this is such a waste of resources, and there are all sorts of issues that we can't address with a meta-analysis. So we need to find a way to take the scientific literature and make it a lot more efficient. We also can't use these PDFs anymore where you can't even, extract the data. How does a meta-analysis work? You have a few coders, just like you did with your, going over the papers, downloading whatever it is that you can, and then going over the PDFs and trying to code those. It's very inefficient. This is not the way that, you can advance things quickly. So we need to also think very carefully about how it is that we, disseminate science, how it is that we aggregate science, how it is that we communicate science to others. And there's a lot of groups in psychology that are working on this right now. We have a big conference called SIPS, called the Society for the Improvement of Psychological Science, where we do hackathons. So we sit together in a round table and we hack solutions. And one of the things that was very promising in the last SIPS is that we are trying to structure what is a hypothesis? How do you communicate an effect size? How do you match between hypothesis and an effect size? And how afterwards this automatically goes into a cumulative meta-analysis that every single point in time will give you the best up-to-date information about the evidence that exists. So lots of avenues and places to go with this.
Yeah, and that has to be very subfield specific, right? Because I mean, in different areas of science, the metadata you actually need is probably different, like so what has to be communicated. So like again, like we have to have like different communities talking about this. We have a couple of people who raise hands. Tim Merrington is here actually, and Michelle Andrade. So Juliana, can you put them in?
Yes.
Tim, hey, good to see you.
Good. How are you doing?
Fine.
So I do have a question, and great. I mean, thanks for the overview of both the work. I'm obviously familiar with both of them. The question I have for you, and it's actually probably a bit more of your viewpoint, is in my experience and in our experience at COS doing the replications is the perceived conflict you can have when you're conducting a replication or even proposing to do a replication with original authors. And then related to that, you guys were just talking about this, which is how other scientists and how the lay public perceives this. And so I'm curious to know two things. One is, have you guys experienced that? So for instance, with undergrads, like what are their thoughts? Do they think it's BS, which is potentially a very dangerous thing to do if you just kind of dismiss a lot of research? Do they, and again, there's like lots of examples of kind of, in my opinion, maybe not the right way to communicate the inability to replicate somebody's results, because then it can create antagonism, which is unnecessary. So I'm just kind of curious about like your perception from both of your backgrounds and both of your projects of like how are they perceiving it, and then over time, what's your perception? And just to give a bit of a relation, there is a project that we're working on with DARPA SCORE. Is actually a really interesting project because we're going back 10 years and we're cutting across even more disciplines. And so it's surprising on two fronts. One is how you still hit that conflict. And I've definitely engaged that a lot with the cancer biology project. But then we also don't. We actually get now a lot more embracing of it in certain areas where they're just like, excellent, great. This is exactly how it should proceed. So I'll stop there and just kind of let you guys comment on that whole line of thinking.
Gilad?
Yeah, it's a good question, and it's something that I, especially this week, discussed following. I saw some reactions to DARPA SCORE and the things that were going on over there, which troubled me with original authors pushing back on the process, but also in so as I've shown, we submit these things to journals, and we've had a lot of rejections. First of all, desk rejections, and then following very hostile peer review that really made me rethink this whole thing about communicating with original authors. And I want to say that at the beginning of the process 2 years ago when I started this, we didn't have any results. This was just an idea, and I communicated to original authors and reached out to them and said, so I'm a first-year assistant professor working with undergraduates in a place in Hong Kong, and we're going to run this on Amazon Mechanical Turk and Prolific. We need your help. First of all, a lot of people didn't answer, and the ones that answered were not supportive. And I have like a list of all sorts of people that started off by thinking that there's no point in collaborating with us because there's actually absolutely no way for us to find anything. This is set to fail from the beginning. And also people questioning the need and the investment of resource in replication and involving undergraduates in this kind of thing. It's like, how can undergraduates do this sort of thing, especially if they come in second year not knowing anything? But it's amazing that every semester, as I disseminate and communicate what it is that we do on Twitter, the more that we submit this to the journals, little by little, we get different reactions. So first of all, there's, I think, differences in the way that, early career researchers respond to this, which is generally, it's like a little bit of confusion. It's like, what does this mean and how can we make sense of this? But a lot of enthusiasm. It's like, it seems like something important is going on. So we want to be part of this. I think at the beginning, we got a lot of pushback from senior researchers, but as, especially the more successful replications that we had, the more careful, the more guides that came out, the more templates, the more that we communicated these things openly and transparently, I think the more trustworthy we were perceived to be. So it's a matter of whatever it is that you do, and I think DARPA SCORE is doing a good job in this. It's just like communicating everything that you do very transparently every step of the way. So starting from pre-registration, I open everything up for open peer review, invite people, whoever wants to come. Every guide is open to see what it is that we do. And I'm very transparent also about the weaknesses. You know, we are doing this with undergraduates. I'm in early career. I don't know what, don't have the answers for everything. So if you're humble, if you're transparent, if you're open, if you try to involve them, then I think there's the potential of things changing. And over time, I think a lot to do with the Center of Open Science and a lot of these ambassadors and people going around and talking about open science, people are changing their attitudes. I can see in my own department, there used to be a lot of criticism and people thinking that I'm stirring up problems in their labs when I'm teaching their undergraduates. All sorts of things like that. But I think recently the tone has changed. At the beginning, they said, okay, you're doing replications, but where is your real science? Where is your real contribution? But now suddenly people are coming and saying, oh, this seems like it's important. Like there's people that are interested, publications are coming out. So it seems like they want to take more part in this. So I think it's a process, it's changing. DARPA SCORE, I think, is going to do so much better than many labs 1, 2, 4. You know, these got a lot of pushback. DARPA SCORE is still going to have a lot of pushback, but not as much as before. And it's going to have a lot of also support and positive responses. For me, it's one of the most revolutionary projects that we've ever had in social sciences.
Yeah, we actually were surprised by, like, we actually have much less pushback than we anticipated initially. I think, well, like, our public image in Brazil is surprisingly good. Like, a lot of people anticipated this, like, oh, we have reduction there. I think we've been very successful in avoiding it, or maybe just lucky, but I think from the start, and I think this is important, we try to picture ourselves, like to sell ourselves as we're not really replicating anybody's work. And I think it helps that we're replicating like individual experiments. I mean, even if this doesn't replicate, it doesn't really invalidate the paper, much less anybody's career. We still have not opened up which papers we're replicating to the public. So, I mean, it's not like we've been doing a whole lot, like we've been going a long way without making this personal. Okay, this is this paper or this is this person in any point. I think this helped. So we've been talking about it for 2 years. We've been gaining traction with like meeting collaborators with her. And like even our replicating labs don't really know whose work they're replicating because we just extracted the protocols, but they're blind to the original studies, which created a lot of problems because then, I mean, a part of the interpretation is on us, and that sometimes overstays. So, I mean, if I had to go back, I'm not sure I'll have done this decision again, but I think this helps. Like, it's very non-personal, and I think it's that helps. We also made an explicit decision from the start not to use, I mean, try to do the replications based on the paper without the help of the original authors. That's different from our PCB and from a lot of other projects. It's not better or worse. I mean, there are different questions, I guess. We figure out that we would not get a lot of useful information because we have very old papers. We have papers from like the late 90s. It's like 98 to 2018. So like, we're not gonna find what was the temperature in the housing facility in 2001. Like, it's not, so we figured that like, we'd get like low response from the authors, that a lot of them would not be able to help because data might have been just lost along the process because these are old studies. And we figure, okay, if we're gonna have like a very heterogeneous response, like only 30% of people or 50% of people respond and only 20% will have useful data, maybe it's just like cleaner to do it without the author's help. That's how we have contacted people. So we have sent out emails to people who have had their experiments included, the corresponding authors, saying, okay, we're doing this. We'll get in touch with you to get some information because we still want the information, just to see like how close we are to the original protocols when we actually, when we're able to know this. Like, we didn't get many responses because like people were not asked to do anything yet, but like the few who had responded, like, oh, cool, nice. I mean, we didn't get pushback from anybody, but it's a very low response rate. But again, I think from the public's perspective, I think we've done very well in terms of course, the results are not out. Like, of course, once the results are out and people realize that, oh, this study did not replicate, we might get into that point. But like up to now, we've been cruising through that. There's an additional issue with the Brazilian Reproducibility Initiative, which is the national thing, which is, okay, we get a number for like, I think the psychology gets into like subfield. Okay, if we do it in psychology and we get to the only 30-something% of papers replicate, this makes psychology look bad among other sciences. We have the country problem, right? Which may be even worse in terms of like the political situation at the moment. So we say like, okay, half of Brazilian science does not replicate. Is this good or bad? I don't know. Like, we don't know the numbers elsewhere. But it can be made to look very bad. And of course, like in a time in which science is very, very undervalued in the country, I do get that this is going to be like a major challenge to communicate in terms of like not saying that this is a Brazilian only problem and not saying that this makes our science worthless. And again, I think we have to sell it as I think psychologists is very at the vanguard of the debate, and like you're very early adopters of like a lot of stuff. And actually, like I think it has made your science more credible, not less. I think we have to make the same argument for like we're doing this, and like no other country has really tried something similar at the moment. So like this put us ahead of the curve in terms of how we're debating and addressing this. But it is a challenge. I think we have not faced it yet, because up to now it's just like it seems like a good idea and everybody likes it. Once we actually get the results out, I think we'll have to think about the things. Michael is somewhere here. Juliana, did you?
At the chat, just at chat.
Oh yeah, he raised his hand, but I don't think he made a question.
Yes.
Via chat.
Acho que ele caiu.
Oh yeah, I don't see he. Well, we have others, right? So there's Clarissa, but she has asked one again, so I'll move and leave her to last. Do you think the Corona pandemic will somehow speed up transition to open science, preprint publication, data sharing, or do you think outside this topic things will remain the same?
Yeah, I don't have a. I'm trying to be optimistic about this and seeing this as an opportunity, but I have a feeling that it's not going to have as much impact as we would like it to have. I think that to us, this really shows to people who are already convinced that open science is good. I think this is like the ultimate evidence that we need open science in order to do things. But I've also heard people that don't believe in open science saying that this is going to be the end of everything because we can't make sense of everything and us sharing it all. I don't know, it's like people find evidence in what it is that they already believe in. Hopefully this will make a change, but it will make a change if really we can show some conclusive evidence in support of this. It's very difficult to do with open science unless you have, randomized and, some consensus about how this helps. I hope so, but I'm not that optimistic. What's your take?
I'll say I'm more optimistic. Of course, like it might not affect open science generally equally, but like at least like in preprint publication, I'm sure it helps because I mean it had already helped in the Zika epidemic. So like 2016 is the year that preprints really took off in biology, and in Brazil at least, where we had like a huge Zika problem, I think that was one of the factors that helped. We had like good case examples of like people like putting a preprint and getting published in Science later and whatever. And so like, okay, so this is possible, this is important. And I think that's
after the pandemic, it stayed this way or it went back to normal, what it was before?
No, I think we gained traction. I think preprints are stably growing, or maybe even more than stably, maybe it's exponentially growing biology since then. I think it gets a lot of people familiarized with the formats. Like even if you don't do it now, you see that things are coming out in bioRxiv and medRxiv all the time. If you're distantly following the coronavirus epidemic, you're at least getting in touch with that. You see that those things actually end up published in Science or Lancet or whatever later. And you see that this is, well, maybe we can do this. I think the exposition to preprints definitely, I think, is helping that. Of course, it also helps. I mean, people who have preconceived notions about it make strong arguments that like whenever a junk preprint comes out, and they will come out because I mean junk science happens, both within and outside the peer-reviewed journals. People will use that as an argument, well, maybe we should reconsider preprints, whatever. That said, I think the debunking of preprints has been much faster than it has it's usually is for peer-reviewed articles. So like my idea is that we're actually fixing things faster. But of course, we have a huge problem in terms of like how information disseminates later through social media. And that's a little bit beyond our control, really, because I mean, things will be used as arguments for pseudoscience, but like peer-reviewed papers will be used for that. Preprints will be used for that, and just like PDFs which are not posted anywhere, such as this Prevent Senior study, will be used for that. Or maybe even like fake PDFs, like somebody does. Like I got all kinds of like complete nonsense in terms of like predictions of like how the pandemic will grow. Like sometimes just a PDF by somebody who works in the housing field who like put up some numbers and like completely meaningless, but like people will share. So I mean that's a little bit beyond. No matter how we decide to publish things. Weird things will get out, and it's controlling controlling that is like a whole different dimension of problems. I think what we can focus is that it is like as a scientific community, we have the best system possible to evaluate evidence, so like to put a stamp of approval or of disapproval in things as fast as we can. I think preprints help in that sense. But of course, how they can be used outside. It is a problem. We can't deny the problem, but it's not been invented by preprints. It will not go away if people stop doing it. So I mean, it's a problem we have to address through other means. In terms of data sharing, etc., could help, but I think then the effect is less clear. I'm not sure. That's actually a very good point. Very good question. I mean, are people opening up data? People are definitely using preprints more during the pandemic. Are they opening up their data more? Not sure. I think it's actually a very good question to address. It might be easy to address, but I don't know. It could be. I mean,
yeah, there's a lot of good work for meta scientists to evaluate what's happening right now and try and estimate how this affected everything.
Definitely, yeah, we have thought about it in terms of preprint dynamics. Are people using them more? We have like. Do we have more authors engaging into this? But like for other open science things, I'm not sure it's going to happen. Next question: is there substantial effort in questioning current methods and proposing changes to avoid the necessity of reproduction studies? This may say, yeah, I fully agree. I think that the point is to get to that. I mean, we want to try to see whether. I mean, one big question here in psychology as well is like whether we could have predicted this from the start, right? Because like, of course, if researchers can predict with like 60 or 70% accuracy which studies will reproduce, then maybe, I mean, we can just focus on the ones that sound likely and just discard the other ones, or or not. I don't know. But that means like a lot of, if we know from the start that things will not reproduce, we know that there's wasted effort, right? I mean, we could have predicted from the start. So I think a large part of it is like, how can you predict it? How can you evaluate it? And how can you make sure that it does not have, like, what should you, if you find good proxies for reproducibility, so like, okay, maybe papers with a bigger effect size or a lower p-value, or which are blinded or which are randomized or whatever, are associated with like more replicable results, you can actually use this as proxies to measure stuff. Of course, like every metric becomes a bad metric once people take it as a target. That's Goodhart's law, but
yeah, I think so. You're working with the prediction markets, and we're working with the prediction markets. Our efforts are relatively small, so you can't generalize from those to everything. But the DARPA score with 3,000 replications, also working with prediction markets, I think is aimed at trying to generalize. So also looking at all sorts of factors. So people typically default to what is the p-value, but now we know the p-values have all sorts of weaknesses. So you look at other things in all sorts of parameters within the original articles, trying to give you what is a better assessor of whether this will replicate or not. So you have two things. You have, these models, machine learning models of, just put in the parameters of an article. But I think also the interesting thing about these prediction markers is also showing that for some reason, and we're not really sure how they're doing this, but people have some intuition about what is able to predict this or not. So the prediction markers by Anna and the Swedish group working on this, they're able to show that people have good intuitions in predicting what will replicate or not, and now trying to model these predictions in order to help us assess not having to reproduce and replicate everything, but also being able to generalize from that.
Yeah, that said, I mean, intuitions are much easier to get in psychology than in basic biology, say. So like I did there's an online quiz for the social science replication project when they just give you the facts. I got 20 out of 21 right. And like I'm just like, but it's my postdoc got like 18. The other guys in the group were like 14. It's like most people, like lay people, and there's data on that, can actually make a judgment. Okay, this just looks incredible, like too incredible. And like those science papers were definitely like, yeah, this can be true. And like that intuition was actually pretty good. So like even without looking at methods, statistics, whatever, just you had a common sense intuition that this seems likely, this does not seem likely. I'm not sure that will hold for our project, like whether this, I mean, very specialized people in the field might have intuition, but like that's going to be like two or three people working in the same field. Like the general biologist won't really know whether drug X actually kills cell Y. So I think we might do much worse than what you have done in economics. Those are hard. I mean, I look at the experiments. For some of them, this mostly for methodological reasons, but like for many of them, I don't have a clue, and I'll never make 20 out of 21 on this, I'm sure. So like I think that might be very subfield specific, and I think like psychologists probably like whatever is like common sense. Is closer to common sense will probably get the best predictions. And concerning the machine learning models, they can work, but again, they'll be focused on like very specific, and they're probably easy to game as well, because like if people realize that like what matters is like people describing blinding whatever, they just say a study is blinded, and I'm worried that the metrics might become just an artificial way of people like making their papers more reliable if we take them too seriously. I think it's good to. These models are great to like gain insights on what matters, but I mean we should be very skeptical of like using this for like actually evaluating people. And
I just want to say, like as a side note, that I think replications cannot be eliminated completely. Replications need to become mainstream and part of the process. So if you publish a paper, there needs to be follow-ups on this. So we need to encourage people. We need to incentivize replicators and give people the opportunity, the career chance, the funding, in order to run replications, not marginalize them outside of academia. So people open up their file drawers, people take the initiative to run replications. So we want to minimize, having to go and revisit everything that we've done since science began till today. But we also want to make replications far more mainstream in order for people to take up the task of running replications, so that we wouldn't have to revisit everything from scratch.
Yeah, and an important question is what's worth replicating as well. So like we get a few comments sometimes like from ourselves from our collaborating labs, like, okay, this is just like so badly described, it's not worth replicating, it's not worth trying. We don't even know what cell. And I mean we're assuming that everything is worth trying. Like even if this is completely, you cannot understand the protocol, that's data, right? I mean, if you just exclude this is going to be like, oh no, we should run to see if we really can't do this. So like we're doing this anyway, but I accept the argument that like you could argue that these are wasted resources, because it's like the original description is so bad that we could have like described this as a failure just like without trying. But I think this is a little bit unfair as well. So I mean, maybe even if it's better described, like the general idea can be done. So we're still doing everything. But I think an important question is like, how do we decide what's really important and worth replicating, and what we have to be sure of this and this and this. So we'll try this out and do it in like a lot of labs. And what maybe just like not as important, or maybe just like it just doesn't sound real, so we might as well not go into it. So I think there's a
kind of the process, and I know this is easier in psychology, but whenever somebody comes to me with an idea that is based on the literature, I say, how about as a first step, let's just do a replication first. And then if this works, then we build on this and do the extra step that you want to take. Many of our replications, I put this up as a replication project that we're doing, but almost all of our replications are replication plus extensions. So while we're doing extensions, while we're expanding the literature, we're building new ideas, but we want to revisit the older findings in order first If something didn't work, if the extension didn't come out as expected, is it because your idea is not good, or is it because the replication failed? So almost everything that I do right now with my students, even if it's a novel direction, I say, let's start with a replication. So replication plus extension. And then if this works out, then we can go for the next step.
And I think biologists do this as a, they commonly will replicate something before going on to like extending something. But like if it fails, people just give up on it. So like this don't really get into the scientific record. And I think that's a big problem. So Clarissa asked whether you would think that more as methodologies become complex, the crisis intensifies. Like it's going to get even worse as methods are more complex. Gilad.
So what will get worse?
As methodologies gets more complex, the reproducibility problem should intensify and should become more complicated.
For sure. Like so this is my intuition, right? I don't have meta science evidence to support it, but one of the first decisions that I set for myself is that I'm going to simplify everything that I do. First of all, we understand that if we do even a simple interaction, but definitely moderation, mediation, multi-level errors going in every direction, we need huge samples for that. We never really took power into consideration as much as we should have. We now understand that we can use larger samples because in psychology we can run things easier. But generally we have a big issue with the sample size that we can run, especially in the hard sciences, the exact sciences. So given the understanding that it's very difficult for us to get large samples, we cannot test complicated models. We need to simplify everything and make sure that our statistics and our models, the way that we design our experiments is set so that it will be reliable. So we can't expect using, I don't know, most of my University of Hong Kong psychology department are neuroscientists. So, you've got 10, 20 at the best people that you can put into the scanner. So with these kinds of samples, you can't really do a moderation, mediation, multi-level sort of insights. Even if you do a very elaborate within design, you really need to adjust your resources and your capabilities to the methods that you use. And you need to understand the implications of what it is that you're using. So complex models require certain things that we now know are very expensive or that we don't have. So for me, simplifying. So I focus on main effects. Probably if there's an interaction, I take much effort into trying to adjust in terms of power. And just generally, I think we're also incapable of understanding, communicating, disseminating complex findings to the public. And I'm not talking about public like presidents or laypersons. I'm talking even practitioners. If we give them something that's too complicated and we don't give like all the steps that came before that, they don't know what to do with it. So my first step, simplify everything, focus on main effects, basic interactions, and work your way up from there in order to adjust the methods and the design to what it is that we're capable of.
Yeah, I generally agree. And of course, like if you go outside of psychology, there's a whole problem, not only like complex models, but like complex methods and machines and gadgets and expensive stuff, right? And of course, at the limit, you have experiments that have been done in the CERN with like 25 kilometers that nobody else can replicate because, I mean, that could only have been done there. So like, if you take like some areas of physics, I mean, it's like, of course, they're very good in like working as community, but really like, okay, this is like the community's answer to this. And like, there's no way somebody's going to spread it because nobody can run the experiment again. So that's like the top scenario. But even in biology, I think there's some very complex methods that like 2 or 3 labs in the road can actually do well. And those are very hard. And again, like I think, but we don't even have data on the simple ones. So I mean, like, let's try the simple stuff first. If this doesn't work, then well, then we have a problem. Like if this works, then maybe we can try like, for many things in biology, it's very hard to run a replication initiative as we are doing because they're much more artsy and complicated, and like you need very, very large skills or resources to do stuff. The other thing is like the more complex methods get, the more complex data analysis tends to get. And so if you go into like functional neuroimaging or like microarray data, there's like so many statistical steps just to get to a result. And like so much like flexibility at each step in the pipeline that like you can use that to like tell any story. I think like fMRI has been the prime example of this. from the fields I'm used to at least. And of course, those are the fields that can perhaps be best, can benefit the most from like pre-registration, which is kind of happening even now, and from like building big consortia that do things in standardized fashions. That was the case in genomics, for example, which has definitely improved its reproducibility record. I mean, you've gone from like studies like 300, 400 people to studies like 500,000 people in terms of like genetic epidemiology. So like that's actually a good example of like a community coming together. Okay, so what we need to find are small effect sizes. There are a lot of genes, so we have like a huge multiple comparison problem. So like, let's do this the right way. And I think it kind of has stuck. So I think we have these good examples to follow where like people realize their methods are complex and find appropriate solutions to it. But I think there's like a lot of communities that have not addressed this as well. I'm not sure I have any more questions here. I have a question for Gilad. So your students are undergrads early in their career, is that it? Like early in the psychology course?
Yeah, so the first course that I run this with is a second year introduction to social psychology. We call this fundamentals of social psychology, where the only thing that they had is basic stats. In their first year, they maybe ran a t-test, or somebody sat with them in to show them, unfortunately in SPSS, how to do very simple things. But when they come into the course, I take them step by step using tutorials, using open guides. I put a lot of the learning on them. First of all, because I realized a lot of things I don't know. So I'm trying to take the humble way out. But also, I believe in their ability to educate themselves. If I give them the direction of where things are and where they can find information, they're actually able to do much better, and they're very good with technology. So they're able to pick up R very, very fast and find all the packages that are able to do all sorts of things. And they're also very good at communicating with one another. So as early as second year undergraduates, we start with this, but the highlight of what I do with my undergraduates is during the fourth year, and this is advanced social psychology. And this is our capstone. So these are people or students that are specifically interested in understanding how research and how academia works. It's not necessarily that they want to be in academia, but they want to understand the scientific process. So it's meant for research projects. Typically what we do, or what we did before, is tell them whatever it is that you want to do, like a thesis, let's see what we can do, and then they write proposals. But I wanted to do things that are very defined, collaborative, on a path to something, and that by the end of the semester, it will be a publishable manuscript built on pre-registration and real data collection. And it's amazing that we've been able to, so at the beginning, I said, if I set unrealistic expectations, if they meet me a quarter way, halfway, then we've done an amazing job. I would say about 70 to 80% of them are able to meet all of my expectations, and about 30% of them far exceed my expectations in taking what it is that I was hoping to do, and taking us into a whole different level, a whole new level of complexity, of openness, of research transparency.
Yeah, I wasn't really thinking about ability. Actually, it's more like engagement. So like my impression is like, so I do a lot of teaching in statistics and stuff, which and reproducibility, which pretty much goes into the same direction. So like, if you, my impression is like, if you do it very early in the undergrad studies, they're just kind of like not that interested, because I mean, it's harder to start with saying we have. It goes with like what Tim said. If you don't even build something, it's very hard to like. People want to first get exposed to science, then they realize, oh, there's all these kinds of problems. So people are very, very open to this discussion and to debating this, and actually have very good insights on what the problems are by the end of their undergraduate studies, maybe the beginning of their graduate studies. So I mean, to me, in like teaching statistics is like the perfect moment to intervene. Of course. It might be a waste of resources, because they first like learn a lot of stuff in terms of like how they do research, and then they learn, oh guys, this is you've been doing a lot of stuff wrong. but it helps me to like people already feel that this is important, that like, yeah, they've managed, I mean, they've realized that like, oh, throwing out these data points is probably not right. And like people are doing this all the time. So like you have more subject to debate this stuff if people are a little more advanced. So we're just thinking about engagement, like how do the early student, like the early course students actually view them, and do they get excited about it? Because I feel it's easy to get people excited about it when they realize that we have a lot of problems. But like if they're just starting on the course, I just wanted to know like about their view. Like, wouldn't they rather be like first believing things and then disbelieving? And second thing, do you think any of this is specific to Hong Kong? Like, I mean, I guess like it's a very specific place and time. So like people have probably better skills in terms of like math and computer and coding and than elsewhere, discipline maybe. So like, do you think that your model actually works elsewhere as well, just as well? Like, I mean, you've been through the Netherlands, you're from Israel, I guess. So like, how do you view this international thing?
Yeah, so actually I'll start from the second one. So for me, one of the reasons why I was spending most of the last year going around the world and finally getting to Brazil in order to present this sort of thing is because we're inviting collaborations where we're hoping that people will take what it is that we've done and at least try it out in different places. I know that a lot of people are skeptical, and then one of the first reactions is, okay, so you were able to do this in one of the top universities in Hong Kong or Asia, but this will never work with our students. And my reaction to this is, let's make this an experiment. How would you know unless you tried? And by now we've gone through all of the difficulties. So we give you ready materials for you to try this out, and there's no harm in trying this out. So don't immediately implement this in everything that you do across the university, but definitely we want to see other people and work with other scholars to pilot this in other places. My initiative is not the only initiative. So if you look online, some of these are in my slides. So you have CREP, and they're very successful. And we're talking about many countries around the world, definitely not just Hong Kong, where you have undergraduates, sometimes in their thesis, or sometimes in their coursework, working on replicating. They're replicating things that are much more recent. They're choosing their replication targets very carefully, and they're more social psychology than judgment decision-making or social cognition. So there's a few initiatives with already enough evidence to show that this is not a Hong Kong thing, and undergraduates can do well. I even know of some initiatives that are trying this with high school students. So it's just, I think it's a matter of communicating openly about the challenges. I think it's, you know, what it is that they can benefit from this, trying to motivate them to do this well, and really structuring everything for them step by step, what it is that they need to do, and moving from this, you know, instructor with a PowerPoint tells the truth to students, moving from that into a model where students take responsibility. So in my postdoc in Maastricht University in the Netherlands, they have something called problem-based learning. Actually, the instructors don't stand in front of a class. The students do everything. So they are the ones that set the learning goals. They are the ones that go out and seek information. They are the ones that come and integrate this. And for me, this is what science is about in the way that we communicate. Now, about how do we get people on board for this? So at the beginning, I have to say, my course is very different from everything else that's done at the university. So the first 2 weeks in the add drop period, where students can drop out, I set expectations very, very clearly, telling them everything, including the scary part. So the students that remain after the 2 weeks and choose to stay in my course and work with me on this are students that have been scared, understood. I even give them a quiz, an exam on the syllabus, so that it's like a contract between us. You know, I tell them what is expected, and they tell me we've read and understood, and we accept this as a challenge. So the ones that remain with me in my class are already seem to be motivated, or at least curious about what is going on and what's happening in this class.
So what's the dropout rate?
What's that?
What's the dropout rate?
It's very high. And because I'm not the only one giving these sessions, so fundamental of social psychology is given by different instructors, and advanced social psychology is given by different instructors. I've had a lot of students come in and complain about how much more hard this is, especially in the first semester where everything was confused and we didn't have a lot of structure and there were a lot of challenges and not everything worked. There was a lot of frustration during the semester, but when the office came to me, the department office came to me and they said, you know, a lot of people are complaining, is everything okay? I told them, let's hold off with this for a while. Let's let the semester end. Let's see the evaluations, then give me another semester. And if at the end of the year, the students push back so hard on this, then we can go back to the original. By the end of the year, the evaluations for this session and how people conclude their experience is a very, very positive experience about this whole thing. And especially, and this might be a Hong Kong thing, I don't know, but it's definitely a cultural stereotype, which is why I try and avoid this kind of conclusions. But there is a lot of competition, and the competition here is about doing well. So if you set the competition and the incentives for transparency rather than finding significant value P lower than 0.05, it's amazing how well they do on transparency. So sometimes we have pre-registrations of like 70 pages, and then we need to like structure this in order to make this more feasible for people to go over this. But by the end of the second semester, the end of the year, when they saw that we're getting media attention from mass media like Psychology Today, the people are reacting on Twitter. We've had the first publication being accepted. So for undergraduates to be able to put on their CV, first of all, that they have a preprint, so with another scholar. And it's not just me, but also early career researchers from around the world. So they've collaborated on a real science project that they can put into their CV. But if it's published in a journal, this is a big boost for them when they're applying not just for academia, but for every job in the industry. So once a word of this came out, suddenly there's a lot more interest, and the students are willing to do a lot more and invest above and beyond the requirements of the course. So I think it's about structuring things up. It's about setting expectations correctly, but also at the end, giving some incentives for them to do well in open science and take part in this kind of initiative.
Yeah, Brazil has actually a very long tradition of engaging undergraduates in research, but it's mostly at the lab level. So people will look up for a lab and start working there. So it's not very well integrated with like the whole course and stuff. So, but I definitely think the model works. Just wondering like what's the best time to do it. But I certainly agree with the thing the undergrads can do stuff. We've worked with crowdsourced projects as well, and I definitely agree that like setting the expectations on transparency is the way to go. I think we have and that's not that hard to do, but I think we have a very long way to go in biology, at least in terms of like valuing this as we should. It's happening, but I think it's just slow. But as institutions, I think we can definitely do much more in terms of like evaluating people on the right things.
Yeah, I just want to say, if anybody is interested in trying this sort of thing, then please reach out to me and I'll share everything that we have and we can talk about how you can do. Even if you're a biology, but you want your students to do some research just to get their hands dirty in something like judgment decision making in psychology, we can work together on this just so you can get them to understand open science and method.
Yeah, I think that's a valid point. I mean, of course, the problem in biology is like, well, it's expensive to do stuff. But I mean, in terms of like science training, I think it's personally reasonable for like people to start like, yeah, like to get trained in data analysis, whatever. I mean, just get like an open data source out of something. I mean, there's a lot of there's a lot of things that can be done as secondary analysis on primary data. I mean, of course, that's not hands-on lab training, but in terms of like stats and analysis and whatever, I mean, there's a lot of projects you can do just with what's out there, which are not as, there could be replications, could be just like new analysis, but I mean, that, and that is free basically. So I think we can go in that direction, even in biology or in expensive fields, right? Do you have any more questions? Yeah.
No.
So yeah, I think we can call this a day, right? I mean, we've been talking for an hour and a half already. It's been really great to meet you. I mean, I'm sorry we can't have the barbecue and the team along. I mean, it's been more like a lonely experience here. Like most people not show up in video as well, so like we don't really see everybody. But we've had a reasonable audience. It's now decreasing slowly, but so that probably means we have to end here. But like, thank you very much for being with us. I definitely think we share.
Thank you. It was a pleasure, and I have a feeling I'm gonna make it back to Brazil once all this craziness is over, and then we can meet in person and do more.
Yeah, and same for us, and you're certainly welcome to come here and meet the team whenever. So this will be recorded online. It should be on our YouTube channel shortly. I think you can probably watch the streaming in Facebook even after this ends, right, Juliana?
Right.
Yeah. So I mean, if you want to like reach out to people and tell them about this, I mean, this is going to be accessible online. Otherwise, both of us, give it much more than us, have sent links and stuff. But if you want to look us up as well, I put up Gillad's on the chat, but our website is reproducibilidade.bio.br. So you're welcome to look us up there. We're in Portuguese and English, and we're easy to find. So thank you very much for being with us. Gilad, thank you very much for accepting this. And hopefully we'll, in a month or two, come back with like more of these debates with other people as well. Thanks a lot.
Bye.
Bye.
Eu finalizei o streaming lá no Facebook, tá?
Beleza, foi ótimo. Aí depois só nos manda o vídeo ali e tal, compartilha com a gente ali pra gente postar no...
Tá, eu compartilhei já o da reunião, aí logo que processar eu passo desse daqui também.
Beleza, obrigado.
Nada.
Você baixou as minhas aulas de estatística lá? Tem que ver se eu botar isso aí no ar ou não.
Baixei tudo. Tá tudo numa pastinha.
Compartilhar no Drive aí e tal pra mim?
Tem, claro. Eu compartilhei já, mas eu compartilho de novo. Tá numa pasta e tem todas as datas.
Me manda esse link aí que eu vou baixar e eu já libero você. Tá?
Mando sim. Pode deixar.
Não. Tá.
Tchau.